Welcome to Changemakers, the future of health. I'm Dr. John Whyte, the Chief Medical Officer of WebMD. Messenger RNA, mRNA technology, can it revolutionize medicine? My guest today is confident it can. Stéphane Bancel is the CEO of Moderna. I had the opportunity to sit down with him at one of their state-of-the-art manufacturing facilities in Boston. He explained why he firmly believes mRNA science can create a new generation of transformative medicines, especially in critical areas such as cancer care and latent viruses such as EBV and CMV. He also revealed why he chose to make rare disease a priority focus. Although we call these diseases rare, collectively they impact over 300 million people worldwide. Listening to him, I was struck by this fascinating perspective of drug development through the lens of someone trained as an engineer. If you're curious about the ever-evolving landscape of medicine and what the future holds for healthcare, this is an interview you need to watch. Stéphane, thanks for joining me today.
Thank you so much for having me, John.
I was fascinated by your website where it specifically says that mRNA could revolutionize. That's the word that you use, revolutionize medicine. And it goes on to say it's all about the protein. Why revolutionize? Too strong a word or mot juste?
So I think it's the right word because if you think about it, since the first medicines were made available more than 100 years ago, it is what I describe as analog medicine, whereas the chemical structure of an aspirin or a Prozac or a Lipitor are all very, very different. But what is really quite remarkable with mRNA is an instruction. It's a piece of code. You send information to the cells, of course, like natural information. That's how, as you know, our body works to make proteins. And you instruct the cells to make the protein or the proteins that you need. And even if a protein is very complex in structure, which could be a limitation in recombinant, well, because your body makes it, then it knows how to do it. And so that's the piece that for us is really a revolution, is the ability to use always the same molecule, the same chemical structure, to send an instruction and then for your cells to do the rest.
Well, let's take that to practice. And really your pipeline focuses on that practice. And let's look at oncology. In oncology, everyone talks about you want personalized medicine, you want precision medicine, less so for overall screening, that's a general population, but you really have made it an approach where you talk about this individualized cancer vaccine strategy. So explain to our audience, what does that mean?
Actually, we used to call it personalized cancer vaccine, and as we thought about it more, for me, a personalized medicine is a medicine that you adapt to my needs, to my mutation. But in that case, what we do is one step further, is we design a medicine just for you. So for example, if you were to have, let's say, a lung cancer, and if I was to have a lung cancer, there's very high probability that the medicine we'll design for you or for me will be totally different. And the way we do it is we start by the genetic information of your cancer cell. So we read the entire DNA. We do the same thing with a healthy cell of your body. And we look at the comparison of every base of the DNA, the 3 gigabytes of information, to figure out where you have mutation. And then we make a product just for you to educate your immune system, your T cells, to recognize the mutation of the cancer cell that they have missed so far, so that your immune system can go to work and get rid of your cancer.
Now, this is a new approach. This is something that has been in development for a while. As it iterates, how do you think vaccine strategy for cancer will look in 3 years, 5 years?
I think we're at the beginning of a total paradigm shift in how we treat cancer. And I not only look at our technology, I'm also fascinated because, as you know, I used to spend quite a number of my career in diagnostic. Mm-hmm. I'm fascinated by liquid biopsy. The ability to find pieces of DNA of cancer cells in blood.
Right. early on, as opposed to imaging, we have to wait till 100,000 cells, 500,000 cells, but this could be fragments early on.
Exactly, very early on. And as we all know, the key with cancer for survival is getting it early. We all know that. And so the ability, think about just pancreatic cancer, which most of the time is a death sentence. If we could find really early on in stage 1 or 2 of pancreatic cancer, and we could do something about it by designing a product just for that patient, you could change drastically cancer care. And I think this is really a few years away between our product that now is in phase 3. It's possible as the phase 2 data matures, because it's already very strong, that we could potentially go for accelerated approval. And the liquid biopsy progress and iteration of those technologies. I really think in a 3 to 5 years time frame, cancer care will look very different and will save a lot of lives.
And this is a way of thinking that other people have tried. Why will you be successful?
Because we have the right technology and we have the capital, but also the right partner with Merck to do it. I don't have to explain to people the role that Merck has had in immuno-oncology. We have the right technology. As you know, John, cancer vaccines have been tried for decades.
Yes.
The success rate is 0%. They all have failed. And so when we saw the data that we shared late last year, and then more at AACR and more at ASCO again, it's working. There's no scientific doubt that this is working. And we talk to the PIs, and you hear the story of those patients that responded to the drug and the change in their life. This is not a fluke. This is not an accident. And so I think what's happening is we have a technology that we can customize, because what the field has tried before us was shared antigen. But because we know now cancer cells have hundreds, sometimes thousands of mutations.
That's what makes it so hard to treat.
Correct. But so if you have to share an antigen like a KRAS antigen or other antigens, you might not do the trick, because we should never forget that the cancer has been growing in your body and your immune system has not been able to take care of it. And so we really believe that this approach, in the current technology, we stitch together, we attach together 36 mutations of your cancer. But we know we need only one that your T cell will recognize for the T cells to do their jobs.
I want to turn to latent viruses, specifically EBV, CMV. I've been a physician a long time. We've been talking about these viruses for quite some time. Why do you think we haven't made progress in effective diagnosis and treatment? And how are you changing that?
That's a great question. I think the field of vaccine makers, as you know, has all tried to do a CMV vaccine. They failed, EBV, they failed. And I think because, again, the technology, going back to your word, revolutionize, the technology allows to follow natural biology. What do I mean by that? The CMV and the EBV and all the latent virus are very complex protein structure. As you are aware, in CMV, one of the antigen that is important for CMV to get into human cells is the pentamer. And as the name says, it's 5 proteins that have to come together in a special structure. And people were not able to make that structure using recombinant. But if we can make the 5 mRNAs coding for each of the components of a pentamer and put it in a cell, then the 5 proteins will be made. We've shown and published that. And what is beautiful about biology is they will assemble to make the pentamer, like the CMV virus does in your body when you get a natural infection. And so we're able with mRNA to make extremely complex protein or antigen to prepare your immune system to be ready for it in case you get natural infection. And that's what I think is a big scientific breakthrough with mRNA to latent viruses, which is why our CMV, which is now in phase 3, it's almost fully enrolled. The phase 1, phase 2 data are very impressive, both GB antigen and pentamer antigen. Same thing with EBV. So I think we have a serious shot at bringing to patients prophylactic vaccine to prevent those infections, and not only the short-term consequences like birth defect with CMV and mono with EBV, but what we're very excited at Moderna about is the long-term complication given to the human health, because those viruses, once in our body, are in our body forever.
And that's the impact of treating these early on, hence why we're calling them latent. So it's a terrific point. We did a tour a little earlier, kind of impromptu when I came in, and we had a quick discussion around rare diseases. And that's what I want to come back to and was very excited to talk to you about it today. And just for reference for our viewers, 7,000 rare diseases, 300 million people impacted worldwide. So we call it rare, but it has a significant impact on people's lives. And only 5% of these 7,000 have approved indications. So I want to ask you, why did you decide you're going to make this commitment to rare diseases? Is it because of the platform that you described early on?
I think the commitment first is to the families and to the kids. I have kids, and it's a thing we've always discussed with my wife since thinking about joining Moderna, when it was just an idea and not much of a company. It was always clear that mRNA, if it was going to work safely in human, could be a great technology for rare disease, because a lot of those rare diseases, John, as you know, are coming from protein deficiencies, because kids have the wrong instruction in their DNA from mom and dad, inside the cells. So if you think about the recombinant technology and what happened with enzyme replacement therapy that has helped so many families, those are proteins that are secreted and basically turning in your body, in our blood. But a lot of disease that are rare disease are basically genetic mutation of protein intracellular, inside the cells.
So you're changing the instruction rather than aiming for replacement.
Correct. So at this stage of the technology, we're not aiming to do gene editing or change the DNA of a patient. We are trying, let's say, if a kid has propionic acidemia, so a protein deficiency inside his or her liver, we're trying to get mRNA IV through a technology of lipid that is different from the one used for the vaccines, for example, to get the mRNA inside the hepatocyte of the liver, to express the protein that the kids cannot make functioning. The animal models have been very, very exciting. And recently, in the spring of this year, we shared that as we were going up in dose carefully for the safety of those kids in the studies, that we're able to reduce by 75% already, including the low dose that most probably in the phase 1-2 were not efficacious, 75% reduction of medical decompensation events. And those kids, as you know, are kept alive by the heroic work of their doctors, nurses that take care of them, and the parents, of course, because every time they have a decompensation event, they might end up, in hospital, they might end up in the ICU, and some might end up dying. And so, very exciting that the technology enables us to basically give them the right protein, regardless of where it is in the body, regardless of how complex it is. If you think about the propionic acidemia for the scientist geek like we are here at Moderna, this is a protein deficiency in the mitochondria of the hepatocyte. So if you think about it, we have to get the mRNA specifically to the hepatocyte. And then once it gets into the hepatocyte, make the protein, which is a very complex protein. It's actually 2 mRNA. And then it has to be chaperoned inside the mitochondria of cells so it can have its clinical impact.
But you've also focused on ultra-rare diseases, which some estimate is literally 1 in a million. And this is these N of 1 trials. And we talked about just my own experience at FDA. How do you prove safety and efficacy when you literally have an N of 1? So how are you thinking about that?
Yes. So we think, again, having this platform technology where we always use the same chemistry for all of those rare diseases in the liver. If you think about the liver, I'm told there are around 1,500 rare disease in the liver, because the liver is such a big protein machinery for the human body. So if you think about our technology, because every product we will make for the liver will have the same lipid to protect from the blood degradation and to take the mRNA inside the hepatocyte. And the only difference between drug 1 and drug 2 will be the order of the instruction, the nucleotide on the mRNA. But the chemical matter we'll give in your body will be the same. So if you think from a safety standpoint, it's a huge de-risking versus the small molecule world where every molecule is a chemically different structure. The other piece is what we're doing in cancer, which is why this technology is quite interesting, how you can connect dots because it's all information-based, which is, if you think about cancer, as we discussed a minute ago, I say that if you had lung cancer, if I had lung cancer, we'll make a different molecule for you and I. So there's already a path with the agency that has been tried in cancer to have the approval not of a drug, but of a process. We think we can prove to FDA that every time we make a product through genetic information, we have the same outcome in the end. That's what we're doing for cancer. I think this technology down the road is usable like with the same way for ultra-rare disease.
I have to ask, just by the way you're explaining all of this, that you've trained as an engineer.
Yes.
And I wonder, because when I've talked to other people interested in drug development, a lot of them are physicians, and it just seems like a different mindset. Do you feel you have a different mindset with your engineer training as how you're approaching these problems and how you're approaching treatments and diagnostic?
I think most probably, as an engineer, I think we are trying to find solutions to problems. And so we always try to design the drug backward. Starting by, okay, what's the biology? What's the...
Reverse engineer.
Exactly. Which is, okay, what's happening for a disease like cancer, the mutation of DNA and the role of the T cells. And then to think about, okay, how can we use this very malleable, flexible technology to do what is required for having the right biological impact. And so I think the engineering mindset has helped us figure out how do you really customize, how can you reuse bricks of technology. It's a bit like a Lego. mRNA is a bit like a Lego where you put different bricks of technology. And the piece I've always loved about it is we make natural protein. Think about, again, we talked about aspirin and Prozac and Lipitor earlier. Those molecules that have helped millions of people do not exist in nature. And so when you go after discovering medicine in traditional pharmaceutical, you do something really hard. You're trying to make a molecule that doesn't exist in nature work in the human body while being safe.
We're in a big building here, lots of buildings. I joked about finding the right entrance, but when you came here in 2011, it wasn't a very large company. You were running a French diagnostic company as the CEO. Why did you choose to come here? And I'd heard from various persons you were employee number 2.
Correct.
So just the second employee. And here, how many employees now?
We're getting close to 6,000. And so the reason for joining actually is quite simple. When I first heard about this idea of using mRNA as a drug, I told the founding team, which was a group of academics from Harvard, MIT here in Boston, and who I think is the best healthcare VC in Boston. My first reaction was, you're crazy, right? This thing is never going to work safely in human. And the more I thought about it over the coming days and weeks, the more it became clear to me that if we could find a way to make this work safely in human, it will change, it will revolutionize, let's go back to your word, medicine.
That's your word.
That's correct. It will revolutionize medicine. It will change fundamentally medicine. And I use the recombinant industry as a mental model, which is if you think about what recombinant was in the 70s with tiny Genentech, tiny Amgen starting, going after this new technology that pharma had no idea how to use. And if you think what, now it's 50 years, what it has done for patients in the last 50 years. And you can start with the growth hormone and the insulin and then the PD-1s and so many other drugs in the middle. It has really changed hundreds of millions of lives. And so what if, there was a thinking in 2011, there will be this new technology that could be potentially more profound because it's an information molecule.
Because you're CEO in another country.
Correct.
You'd have to come, not around the world, but to another country, bring your family with a very small company, as we said, number 2.
Yeah, and it was just this idea that it will change medicine forever. I told my wife I would rather take a 5% chance to build a company that will change medicine forever in a very profound platform-like way than taking a 90% chance to build an average biotech company that maybe might get one drug to market in the next 20 years. And so that was the bet. It was a bit crazy. All my friends said, don't do it. And I still did it. And I'm very thankful because it has been, in the last 12 years, an incredible journey. Of course, with COVID, this is something that none of us, of course, ever planned, a pandemic. It has been remarkable to see the commitment of the teams, the teamwork with our partners. It has been an incredible journey.
Well, let's talk about COVID. And behind us, here we have all these vials that represent the map. Has COVID vaccination changed how we think about drug development?
I think it has in at least one obvious way, which is showing that when there's a big medical need, if we all come together, different players in industry, because we could not have done it alone, and regulators and government officials, because, for example, the NIH had a big role, as you well know, and the CDC, we could do extraordinary things. I think if you talk to any leaders in any of those parts of healthcare in, even December, 2019, and you tell them we collectively can get several high-efficacy vaccines in less than 12 months to a finish line by still doing the phase 1, phase 2, phase 3, nobody would have believed it. And because there's such a huge challenge, as I like to tell our teams, back then when we started running after the virus, we're at war with this virus. It's literally killing thousands of people every day. And every day matters, every hour matters. And the energy, the commitment, and it was personal for everybody. What is fascinating, I used to come here to Norwood regularly. Sometimes I will come visit in the morning, and I will have engineers that had been here all day the day before, worked through the night, went to the locker room, take a shower, were still working. And for everybody it was personal because it was kids locked at home, a lot of time, spouse not working, worried about parents. Many of us had parents or uncles and aunts, that got hospitalized, and some died. So it was very personal, and the collaboration was amazing. I remember sometime we had 2 or 3 meetings with FDA in the same day, which will never happen in normal times. But again, and sometimes it will be Saturday night, and the energy and the commitment of so many people is what made it possible.
Did it revolutionize medicine?
I think it was the first step in revolutionizing medicine in terms of, 1 year to launch versus, as you know, the fastest vaccine before was 4 years to launch. So if you think about what the world would have looked like for another 3 years with a pandemic, of course, it will have, slowed down as we saw in 1918 with more and more people infected and natural immunity, and unfortunately, a lot of people dying. But it's the beginning of a revolution. I think it has shown people that this technology can change everything. And that's what we do believe at Moderna. It will change everything.
We're starting to see cases increase right now. There's talk about whether people need boosters in the fall. What's your thoughts on what people need to know today?
I think a few things is that COVID is here to stay with us, we believe, forever, like flu. We don't believe this virus is going to disappear in a year or 6 months or a few years. It keeps on evolving, like flu. I think flu is a very good mental model for people to have who don't necessarily understand the science like you do. And we need to adapt the vaccines to where the virus is and is evolving. That's what we have done. FDA has been very clear in the spring with a new strain of COVID, the XBB that they wanted. We worked on it all summer. We have tens of millions of doses in a warehouse ready to go. We're waiting for the FDA green light to be able to ship them to pharmacies and to the CDC recommendation of who should get them. But I think that the mental model that doctors should have, healthcare professionals and consumers, which is you need to protect yourself. And I think we all have a civil responsibility to protect each other.
But how do we battle this COVID fatigue that we hear from people, as well as the misinformation? What's your counsel there?
Yeah, so I think on the COVID fatigue, as you know now, there's been data done in terms of clinical study that it is safe to get a flu shot and a COVID shot. So one of my personal recommendation is if you get a flu shot, get the COVID shot in the other arm the same day. It's safe. It has been shown, CDC recommended. And I think over time, one of the cool technology we are working on is to combine into a single shot COVID and flu. We're also working on RSV. As you know, we filed to the FDA an RSV vaccine that I hope will be approved soon. The data is very, very strong. And we also want to combine this into a single dose. So ideally, in a couple of years, you go to your local CVS and you get your COVID, your flu, and your RSV in one shot. and you spend a nice winter. You don't worry for your health. You don't worry about other people. And that's what we want to do at Moderna with this technology, is make your life easier and protect you.
If I went around and asked the other executives, what's Stéphane's leadership style? What would they tell us?
I think they will tell you he's very mission-driven. There's not a meeting, I think, that I don't point to the wall. In conference rooms, we have a company's mission painted on the walls to remind people that we are here to maximize the impact of this technology and this science to help as many lives as we can. I think they'll tell you he's really impatient, because for me, every day we waste where we don't do our best work, maybe in 2 months or in 6 months or in 5 years, somebody will get hurt because we didn't do our best work, and that's on us. And I like technology, I like innovation. You know, I'm a science geek, tech geek. And so I like to find new ways, new technology, new invention.
You took a chance in 2011. You say it was a 5% chance, but you wanted to make an impact and revolutionize medicine, as we've discussed. And in 2022, you were given the Chevalier, a Knight of the Légion d'honneur, which is France's highest distinction of military and civic accomplishment. How'd that make you feel?
Very humbled, because first, every time I got recognized in the last year or two post-COVID, I've always told whichever organization was giving me a prize that it had to be for the team, because I didn't do the COVID-19 vaccine by myself. We are literally hundreds of people and then into the thousands, each playing a very important role, because as you know, making medicine is like team sport. But the piece that was humbling is, in a country where we like to see more entrepreneurs in France, I think having an entrepreneur recognized for contribution to society, I hope is a good example to give to the next generation of leaders, engineers, doctors, scientists, the courage sometimes to go and do this big leap of faith, which I did. It was a crazy idea using mRNA to inject in people. There's a lot of amazing science happening every day around the world. And I really hope that we have more entrepreneurs turning those into products so we can help a lot of people.
Now, should I have been calling you sir?
No, I love this about the US, the informality, so Stefan is perfect.
Well, Stefan, thank you very much for taking the time today.
Thank you so much, John.